Good morning, students. Welcome back again. We have been discussing renal physiology. Today we will see the structure of a nephron. We will differentiate between cortical and juxtamedullary nephrons. We'll see the basic process of urine formation and explain the first step in urine formation, that is glomerular filtration. We'll see how the filtration barrier is formed, and we'll define glomerular filtration rate, which is also referred to as GFR. In the next lecture, we will enumerate and explain the determinants of GFR and describe the renal blood flow and its effects on GFR. So this is the structure of a kidney. It is composed of an outer cortex and inner medulla. The medulla is composed of pyramidal shaped structure, which is called renal pyramids. And in the cortex as well as in the renal pyramids, special structure is located, which is called the nephron. So what is the structure of a nephron? We will see that. The yellow structure over here is called nephron. Nephron is the basic structural and functional unit of a kidney. So this cup-shaped structure is called the Bowman's capsule. Extending from the Bowman's capsule is a convoluted structure which is called the proximal convoluted tubule. Then we have a U-shaped structure which is called the loop of Henle, and extending from the loop of Henle is another convoluted structure which is called the distal convoluted tubule, and then we have this collecting tubule and the collecting duct. So this is a tubular structure. This whole is a tubular structure, and it is meant for collecting the components from this artery and formation of urine. Okay. The loop of Henle is further divided into two parts. This portion is called descending portion, and this is called the ascending portion. Descending portion is further divided into two parts, the thick portion and the thin portion, and the ascending portion is also divided into thin portion and thick. So this is called as thick ascending limb of loop of Henle. Again, the same structure of a nephron. Over here, we will also discuss the arterial supply of the nephron because it is important in the process of filtration and formation of urine. Kidney is supplied by a renal artery that divides into segmental arteries. Segmental arteries give rise to interlobar arteries. Interlobar arteries are further giving rise to the arcuate arteries, and arcuate arteries give rise to smaller branches which are called the interlobular arterioles. From the interlobular arterioles arises a very important arteriole which is called as afferent arteriole. So over here you can see this structure. This is called as afferent arteriole. Afferent arteriole enters into this space and give rise to a capillary plexus which is called as glomerulus. From this capillary plexus, another arteriole arises that is called as efferent arteriole. This efferent arteriole give rise to another capillary plexus which is called as peritubular capillaries. The peritubular capillaries surround the renal tubules. These peritubular capillaries are also forming a very special structure which is called as vasa recta. As you can see, this vasa recta is forming a U-shaped arrangement around the tubules. Okay, the peritubular capillaries then further form the interlobular veins, arcuate veins, and ultimately drain into the renal vein. Now we will see the difference between two types of nephrons, that is the cortical and juxtamedullary nephron. So we have actually two types of nephrons within the kidneys. One is called the cortical nephron and the other is called the juxtamedullary nephron. As you can see that this is the structure of the kidney is divided into outer cortex and inner medulla. Medulla is further divided into an outer zone and an inner zone, which is a deeper zone. Okay. So the cortical nephron is mainly located in the cortex. As you can see here, it is located near the outer cortex, while the juxtamedullary nephron, juxtamedullary nephron is located near the medulla. The Bowman's capsule, remember that the Bowman's capsule, the proximal and the distal convoluted tubule, they are located in the cortex but near the medulla, while in case of the cortical nephron, this is located near the outer cortex. Okay. Second difference between the cortical and juxtamedullary nephron is that the loop of Henle in case of cortical nephron is shorter as compared to the juxtamedullary nephron, in which the loop of Henle are quite long and extending deep into the medulla. The third difference is that the peritubular capillaries, which are present around the tubules, are forming a special arrangement in case of the juxtamedullary nephron, which is called the vasa recta, which is absent in case of the cortical nephron. And this special vasa recta, this has an important role in the maintenance of concentration of urine. We will see later on what is that. So these are the major differences between the cortical and juxtamedullary nephron, as you can see here. So what is the basic process of urine formation? As we all know that the main function of nephrons and collecting ducts is to control the composition of body fluids and remove the waste from the blood in the form of urine. So urine contains waste and excess water and electrolytes that should be removed from the body. And this process is accomplished by three main processes. Number one is called the glomerular filtration. Then is the tubular reabsorption and tubular secretion. We will focus on these processes one by one. The first process is the glomerular filtration. What is glomerular filtration? It is simply the filtration of fluid from these capillaries into the Bowman's capsule. Okay. So we will see, first of all, we will see basically one by one what is filtration, what is reabsorption, what is secretion, and what is excretion. Over here you can see a very simplified version of the nephron. This is the Bowman's capsule, all the tubules over here. Okay, and this is afferent arteriole, efferent arteriole, the peritubular capillaries, and then the veins. So what is happening here? We will discuss the process of filtration. What is filtration? This is simply the process in which fluid is filtered from the afferent arteriole or from the glomerulus into the Bowman's space over here, into the Bowman's capsule. This process is called filtration, and because this is taking place under pressure, so this is called as ultrafiltration. The process of filtration of fluid from the glomerulus into the Bowman's space is called as filtration. Okay. So in this, the fluid is coming from these capillaries into the Bowman's capsule. Okay, now you have also seen that small amount of fluid has filtered while the rest of the fluid has moved from the efferent arterioles into the peritubular capillaries. What is going to happen with this is that from this filtrate, some of the substances and fluid is going to be reabsorbed into the peritubular capillaries. The process is called as reabsorption. And further, some of the substances are going to be secreted from these peritubular capillaries into the tubules. That process is then called as secretion. And the rest of the thing that is left here is excreted in the form of urine. The process of filtration has to take place through a very specialized structure which is called as filtration barrier. Again, we will see that over here, this is the afferent arteriole. This is forming a network of capillaries which is called the glomerulus, and from this arising another arteriole which is called the efferent arteriole. Over here you can see the Bowman's capsule has two layers. One is the peripheral layer and the other is called the visceral layer, which is surrounding the capillaries. The peripheral layer and the visceral layer. The visceral layer is formed of very specialized cells which are called as podocytes because they have finger-like foot processes. Okay. Over here you can see that the capillaries are surrounded by specialized cells which are called podocytes. If we see the enlarged view of this is the enlarged view. Again, here is the capillary endothelium which is surrounded by the basement membrane and the podocytes. You can see that the capillary endothelium has fenestrations. So this is a fenestrated capillary, and the podocytes have these spaces between them. The smaller spaces are called as slits or filtration slits. So these three layers are actually forming the filtration barrier: the capillary endothelium with fenestrations, the basement membrane, and then the epithelium, the podocytes, the food processes of the podocytes with filtration, slits or slit pores. These three things are forming the filtration barrier. So whatever the fluid is being filtered from the capillaries into the Bowman's space, this has to pass through the filtration barrier. What is the importance of this filtration barrier? The filtration barrier is important because it allows only selective substances to be filtered through the arterioles into the Bowman's space, and it does not allow the larger particles to be filtered like proteins. So proteins remains inside the blood and are not filtered into the Bowman's capsule. The smallest protein is called the albumin, and albumin has a size of about 6 nanometers. And these slits or the filtration pores or the filtration slits have a size of about 8 nanometers. So, how a protein with a size smaller than the size of these filtration slits is not passed through the filtration barrier? The reason is the negative charge on the filtration barrier. The capillary endothelium, the basement connective tissue of the basement membrane, and the podocytes are all negatively charged, and the negative charge is going to repel the negative charge of albumin. So this negative charge is very important to prevent the filtration of albumin from the filtration barrier. So the ability of a solute to penetrate the glomerular membrane depends upon the molecular size of the particles and the ionic charge. The particles who have a molecular size more than 8 nanometers will not be filtered, and the particles who have a negative charge will not be filtered. So how we are going to define the glomerular filtration rate? Up till now, we have been discussing the simply about the glomerular filtration. Now, what is glomerular filtration rate? So, rate at which plasma is filtered into the Bowman's capsule per minute, or we define it as the volume of fluid filtered from the plasma into the Bowman's capsule per unit time. So per day, the glomerular filtration rate is about 180 liters, and in a minute, it is 125 milliliters.